Okay, slide is visible or not?
Yes, sir.
Okay, so today we'll talk about the thirst center. Okay, the first one is thirst centers, thirst satiation. We'll discuss it. So the first one will be thirst center. Actually, mechanism of thirst, it is very complicated because there are some theories which are suggesting that this is causing thirst and this is the satiation. Okay, but there is no clear-cut evidence, or we can say there are so many theories. Okay, so you have to remember that one. Okay, so thirst center, it is located in the hypothalamus. I've already told you regarding osmoreceptors also we have talked about it. Okay, so this thirst center, it is located in the anterior part of the hypothalamus. Okay, and in that area there is lamina terminalis. Okay, lamina terminalis is there in the brain, or we can say in anterior hypothalamus. Okay, and some parts of this lamina terminalis, okay, they are considered as the thirst center. Thirst center. So in the first diagram, you can see this is the thirst center, just above the normal osmoreceptor. This osmoreceptor we have already talked about it in the last class. Okay, so this is controlling ADH secretion. We have talked about it. Okay, so just above or anterior part, we can say there is an area of lamina terminalis. Okay, some parts of lamina terminalis, and this area is considered as the thirst center. Okay, so this thirst center, it is divided into three parts. We can say these three parts, it is said that they are involved in the sensation of the thirst. Number one is subphonical organ. Okay, in short, SFO. So if you see the second diagram, okay, this is the subphonical. You can see this is the subphonical organ. Okay, the second one is the organum vasculosum lamina terminalis. That one also we have talked about this one already when we talk about the ADH. Okay, and the third part is the median preoptic nucleus, MNPO. Okay, so these are some parts of the lamina terminalis. Don't think that all the lamina terminalis, all parts of lamina terminalis, they are the thirst center. Okay, it is not like that. Only some parts of the lamina terminalis which are located in the anterior hypothalamus, they are considered as the thirst center. And this thirst center, they are divided into three parts: SFO, MNPO, and OVLT. Okay, and you already know that this one, it will, what to say, be innervating the paraventricular nucleus, supraoptic nucleus, that we have already discussed in the last class. Okay, so these are the three regions or the three subdivisions of the thirst center. So in the thirst center, there are osmoreceptors. Just like osmoreceptors we have already discussed in the last class, which is important for the ADH secretion. Okay, so in the thirst centers also, okay, in the OVLT and SFO. Okay, here you can see the diagram, osmoreceptors, they are also present in the OVLT and SFO. These are the thirst centers. Okay, these are thirst osmoreceptors. Osmoreceptors, you already know what are osmoreceptors. They are the receptors, or the, we can say, nerve endings or nerve which can detect, what to say, changes in the concentration of solute or osmolality or osmolarity. Okay, so in the thirst centers also, these osmoreceptors, they are present there. Okay, but here another important point is that the osmoreceptors which are present in that thirst center, they are different from that of osmoreceptors which is regulating ADH secretion. Okay, so in the last class we talked about some osmoreceptors, okay, which are important for ADH secretion. Okay, but the osmoreceptors which are present inside that thirst center, they are different from that of those osmoreceptors which are responsible for ADH secretion. Okay, you just remember this one. Okay, osmoreceptors, different types of osmoreceptors are there. It is there in the kidney also. Okay, can anyone tell me what are the names of, what is the name of, let's say, osmoreceptors in the kidney? Can you tell? Oh, macula densa cells. Okay, that is also innervated with the nerve. Okay, macula densa cells, we have already talked about it because they can detect the concentration of solute in the glomerular filtrate. Okay, so here osmoreceptors, they are also present in the brain. Okay, so here you have to remember that Kar's osmoreceptors and osmoreceptors for ADH secretion, okay, they are different. Okay, so what are the stimuli through thirst? Okay, what are the stimuli through thirst? Number one, it can be stimulated by intracellular dehydration. Intracellular dehydration. This is caused by the increases in the effective osmolality or osmolarity of the extracellular fluid, or we can say hypertonicity or hyperosmolality or hyperosmolarity. Intracellular dehydration. Okay. So in this condition, there is increase in the solute concentration in the extracellular fluid. We can say extracellular fluid means in the plasma as well as in the interstitial fluid. Okay, when there is increase in the concentration of osmotically active substance such as sodium in the body fluid, okay, extracellular fluid, this will stimulate thirst. Okay, it will induce the sensation of thirst. Okay, so this is very clear. If you take higher amount or large amount of sodium chloride, this is a good example. Okay, after some time you'll be feeling thirsty because you are adding more osmotically active substance to the extracellular fluid. Okay, so this is one stimulus, okay, for inducing thirst. Number two, intravascular hypovolemia, which is caused by losses of extracellular fluid. Okay, intravascular means inside the blood vessel. Hypovolemia means decreased blood volume. Okay, when there is decreased blood volume, okay, which may be because of loss of the extracellular fluid, like bleeding or hemorrhage. Okay, in that condition also the blood volume will decrease. Okay, this can also stimulate thirst center. Okay, so that it will induce sensation or a desire to drink water. Okay, so let's see what is the intracellular dehydration. I told you that intracellular dehydration means increase in the osmotically active substance in the extracellular fluid. That means hypertonicity or hyperosmolarity or hyperosmolarity. Intracellular dehydration, the reason you already know. This is because of increase in the solute concentration, osmotically active substance. Okay, this one we have already discussed also. Okay, so as you know, intracellular dehydration, it is caused by increases in effective osmolality of the extracellular fluid. Okay, hypertonicity or hyperosmolality. Okay, cellular dehydration acts via an osmoreceptor mechanism in the hypothalamus. Okay, so osmoreceptor in the hypothalamus, they will be stimulated. Okay, that is that thirst center, thirst osmoreceptor. Okay, increased blood osmolality is probably the most important homeostatic signal for drinking in everyday life. Okay, so this is very important. Okay, why we used to have a desire to drink water? Okay, it means that the most important one is that there is increase in the blood osmolarity or osmolality. Okay, osmotically active substance they are increased in concentration. Okay, that is why we used to have a desire to drink water. Okay, although when there is loss of blood, that may induce the thirst also, but that is not a common thing. Okay, it is not common at all. Okay, but the most common reason why we used to drink water, or why we used to have this desire to drink water, is that because there is increased blood osmolality or osmolarity. Okay, why this is happening, we'll see. Okay, increase in extracellular fluid concentration of non-permeable osmolytes. Okay, increase in extracellular fluid concentration of non-permeable osmolytes such as sodium causes osmotic movement of water from the cells. Okay, suppose there is increase in the concentration of non-permeable osmolytes, for example, sodium. Okay, so this will cause osmotic movement of water from the cells. Let us say this is the Thars osmoreceptor. Okay, this is the osmoreceptors of Thars center. Okay, which may be in SFO or OVLD. Okay, so you see, this is the osmoreceptor, Thars osmoreceptor. So suppose here outside of this nerve fiber, it is extracellular fluid. Okay, these are the interstitial fluid. Okay, here fluid are there. So we know that this sodium, it has increased in its concentration in the interstitial fluid, or we can say in the extracellular fluid. Okay, so sodium concentration has increased because of dehydration, or maybe because of increased intake of the sodium. Okay, concentration has increased. But as you can see here, this sodium is non-permeable osmolyte. Okay, it is not permeable. That means the concentration outside, it will be very high. It cannot enter the cell, this osmoreceptor. So they are accumulating outside of this neuron or osmoreceptor. Okay, then you already know that what will happen is that if this concentration or sodium concentration has increased outside of the cell, it will lead to osmosis of water. Okay, water will move out of the cell. Here you can see water will move out of the cell. Okay, see that water is lost from the cells. Okay, because water will always try to move from higher concentration to lower concentration. Okay, so you see sodium major determinant of the effective osmolality of the extracellular fluid, because its concentration is high and it acts as if it is restricted from entering the cells. Okay, so it is non-permeable osmolyte. Okay, sodium is non-permeable osmolyte. It cannot enter the cell. So that is causing osmosis of water. Water will come out of the cell or the osmoreceptor. Okay, so in contrast, for example, urea, glucose permeates cell freely and does not exert an osmotic force if it is elevated in either compartments. Thus, hyperosmotic solution of glucose and urea do not increase water intake, but hypertonic solution of sodium chloride or sorbitol do. Okay, so this is very important. Okay, urea and glucose. Okay, here example I've given this glucose. Okay, urea and glucose, they can easily move from outside to the inside, or from inside to the outside also. Okay, so there is no problem regarding their movement through this Thars osmoreceptor. Okay, so that is why they are not responsible for the induction of the thirst. Okay, but you see, sodium chloride as well as sorbitol, they cannot enter the cell, but accumulating outside of the cell, and they are causing osmosis. Okay, that is why they induce the thirst. Okay, so you know very well. Okay, when all of you know glucose. Okay, so when you do what we say football match and all. Okay, they are also used to take this glucose juice. Okay, so when you drink glucose juice, your it will not induce any thirst. Okay, means there is no problem because glucose can move from inside of the osmoreceptor cell to the outside or from the outside to the inside. Okay, but suppose you take sodium chloride solution instead of glucose solution. Okay, you know that you drink 1 or 2 glasses. Okay, but after some time you know that there will be increase in the intensity of that thirst again. Okay, initially it may satiate or suppress the thirst because of that we will be discussing afterwards. Okay, but after some time it means that you are adding more sodium to the bloodstream again. Okay, so again the same thing it will happen here. Okay, more accumulation of sodium will take place near the first osmoreceptor. It will cause again osmosis of water. Okay, so depending upon what kind of solute you are ingesting, okay, depending upon that one, it may induce or may not induce the thirst. Okay, but we know very well that sodium is an osmotically active substance which induce thirst if they are accumulating or if their concentration has increased in the extracellular fluid. Is it clear or not this point? Okay,
Yes, sir.
next. The addition of an impermeant solute, for that is the sodium I've already given you, to the extracellular space causes net intracellular fluid volume depletion and creates a hypertonic state intracellularly. So actually because of the osmosis of water, okay, again loss of water is there, but inside of this osmoreceptor, okay, there may be increase in the solute. Not because there is addition of solute to the inside, but because of loss of water. Okay, hypertonic state may also be developed inside of the cell. Okay, we all know that water distributes according to the amount of impermeant solute in its compartment, the intracellular fluid and extracellular fluid. Okay. So the normal relationship between the increase in serum osmolality and thirst is linear. Okay, serum means, let us consider plasma. Okay, serum osmolality and thirst is linear. What does it mean? It means that if there is linear increase in the concentration of osmotically active substance in the plasma, okay, the intensity of thirst will also be increased. Will keep on increasing. Okay, it means higher the osmolarity of the serum or the plasma, and more will be the intensity of the thirst. Okay, it is linear. Okay, more concentration of solute in the blood means more intensity of the thirst. Okay, but if the plasma is dilute or osmolality is low, that means thirst will also be low or it may not be felt at all. There may be no thirst. Okay, if there is dilution of the plasma. Okay, so this point I've already told you. However, the set point for the osmoreception regulating AVP release is lower than that for thirst. Okay, first I have a question. What is the full form of AVP? Anyone can tell me? Can anyone tell me what is the full form of AVP? Anyone listening or not? What is the full
Arginine
form?
vasopressin.
Okay, arginine vasopressin. What is the other name of arginine vasopressin?
So ADH.
Yeah, it is ADH. So don't get confused, okay? Sometimes I may say that it is ADH, sometimes I may say that it is AVP, okay? So this is a very important point. I've already told you. The set point for osmoreception regulating AVP release is lower than that of the thirst. It means even before you feel thirsty, your body may be subjected to dehydration already. Okay, so you may not feel thirsty, but at that time, the dehydration has already taken place. So because of that, at that moment, ADH release has already been initiated. Okay, that is why it is saying that osmoreception regulating, set point for the osmoreception regulating AVP release is lower than that for the thirst. Okay, so when we can say that when the blood volume is, when the plasma is still somewhat diluted, okay, but dehydration has already taken place. In that condition, ADH release will be initiated already, but you will not feel any thirst. Okay, thirst center will not be stimulated. That is why, okay, set point for AVP release is lower than that for thirst. Okay. Thus, the initial physiological response by the body to dehydration or to increase serum osmolality is to release ADH. Okay. Thirst is a later adaptation to dehydration. Okay. So thirst will appear afterwards, after ADH release. Okay. So the second stimulus for thirst sensation, it is intravascular hypovolemia. So in the subsequent slide, we'll discuss this in more detail. So you already know that intravascular hypovolemia means decreased blood volume, okay, which may be due to losses of the extracellular fluid, okay. It may be because of diarrhea, it may be because of bleeding, okay. Bleeding may take place, okay. You are losing the blood, okay. That can stimulate the heart center. Okay, hypovolemia, that means low blood volume is sensed via the low pressure baroreceptors in the great veins and right atriums. Okay, right atrium. Hypovolemia, that means decreased blood volume. It can be sensed through low pressure baroreceptor. Baroreceptors, you already know, they are the pressure receptors which can detect changes in the blood pressure, okay, which may be in the vein, which may be in the artery, carotid bodies, aortic bodies, we have already discussed. Okay, so when hypovolemia is there, baroreceptors which are present in great veins and right atrium, they will be stimulated. Okay, can you tell what are great veins? Anyone can tell me great veins?
Vena cava.
No, vena cava. Okay, two vena cava are there. Okay, good. So what about hypotension, low blood pressure? Is sensed via the high pressure baroreceptors. Okay, here in this case, it is a high pressure baroreceptors in the carotid and aortic aorta that provide the sensor for this input. Okay, actually this blood pressure and heart fluid balance, all these things, they are all interrelated. You cannot separate it. Okay. So angiotensin II likely contributes during more severe degrees of intravascular hypovolemia and hypotension. That we'll discuss afterwards. Actually, we have already discussed earlier also. Okay. So when we talk about thirst also, okay, we cannot exclude that renin, angiotensin, aldosterone system. All those things, they are also involved. Okay. Now, let us discuss in brief thirst mediation. So as I told you in the beginning of the class, I told you that this thirst mediation, how it is stimulated, how thirst center is stimulated. Okay, there is no clear-cut, explanation. There are so many theories out there. Okay, so the first theory is that hypothalamus mediated thirst. Okay. So thirst osmoreceptors are sensory receptors that detect changes in osmotic pressure, plasma osmolality. So you already know that just like the osmoreceptor for ADH, okay, release. Thirst osmoreceptors, they are also sensory receptors and they detect changes in osmotic pressure. That means plasma osmolarity. So as you already know, they are found in the OVLT and SFL of lamina terminalis of the brain. These are known as I told you, heart center. Okay, osmoreceptors, they are present in the heart center. Okay, so let's see. what happens when there is high plasma osmolarity or osmolarity. Suppose there is increase in the sodium concentration or osmolarity in the plasma. Okay, so this will stimulate Hars osmoreceptor of OVLT and SFO neurons. So as I told you, in the interstitial fluid which is surrounding the Hars osmoreceptor, there will be high concentration of sodium. And you know that osmosis will take place from the osmoreceptors with the interstitial fluid. Okay, so that we can say that osmoreceptor in the OVLT and SFO neurons, they are stimulated. Okay, so after that, as you can see from here, this MNPO. Okay, MNPO, that one is a part of the heart center. Okay, it will also be stimulated, and it is said that this is causing the heart sensation. Okay, stimulation of MNPO, median preoptic nucleus. Okay, neurons, when it is stimulated, it is said that we used to feel thirsty. Heart sensation will be manifested. Okay, but as I told you, this is just a theory. Actually, there is no clear evidence regarding whether this is exactly causing the heart sensation. Okay, but scientists are assuming that stimulation of NNPO neurons is causing heart sensation. Okay, so that is the first theory. Okay, so number 2 is renin-angiotensin aldosterone mediated heart. This you already know very well. Okay, we have already discussed. Okay, so the RAAS is a complex homeostatic pathway that deals with blood volume, plasma osmolarity, blood pressure. Okay, so suppose, let's say there is high plasma osmolarity, concentration of solute in the plasma has increased. Okay, just like the previous theory we have discussed. What will happen is that we know that this osmolarity we have already discussed, it stimulates macula densa cells. Okay, macula densa cells of the kidney, these are the, we can say, osmoreceptors only, peripheral osmoreceptors. And you know that this we have already discussed when we talk about juxtaglomerular apparatus. Okay, you know that when macula densa cells, when they are stimulated because of high osmolarity of the glomerular filtrate, it will stimulate juxtaglomerular apparatus to release renin. You already know renin. Okay, so I have a question. What is the function of that renin? What will renin do after it is being released from the GL? Anyone can tell me? Are you listening or not? Class is being attended or not? Okay, what is the function of renin? Go to hotel, I mean by immediate effect. Okay, there are so many steps to increase the blood pressure. What is the immediate effect of renin? It will convert what?
Angiotensin to angiotensin 1.
Yeah, very good. Angiotensinogen 2, angiotensin 1. Okay, renin enters the bloodstream and renin converts angiotensinogen into angiotensin 1. Okay, then angiotensin 1 will be converted into angiotensin 2 by what? Angiotensin 1 will be converted into angiotensin 2 by what enzyme? Anyone can tell? Or not? SCE, angiotensin converting enzyme, which is mainly coming from the lungs. Okay, now we have angiotensin 2. Okay, so actually this angiotensin 2 acts on the heart center in the brain to cause a sensation of heart. Okay, so we know that angiotensin 2 is very important in controlling the blood pressure, controlling the concentration of solute inside the body, because it has increased the sodium reabsorption. All these things stimulate the sympathetic nervous system that in RAAS system we have already discussed. So one function of angiotensin 2 is that it is said that it stimulates that heart center to have heart sensation. Okay. So these two are the two theories. Okay, for induction of heart. Okay, now heart satiation. So we all know that when we feel thirsty, okay, if water is available, we used to drink water. Okay, so then immediately within a few seconds, okay, within seconds only, few seconds only, you will feel satiation, or your thirst will be suppressed, or it will be removed. Okay, how this is taking place, we will discuss. Okay, thirst satiation. Osmoregulation was initially envisioned as a single-loop negative feedback system in which dehydration increases plasma osmolarity, thereby provides a stimulus for thirst, whereas water intake serves to decrease plasma osmolarity back to normal, thereby causes satiation by removing the excitatory signal for thirst. So this is very simple. That means, suppose you are feeling thirsty because of increase in the concentration of, let's say, increase in the plasma osmolarity. Okay, you are feeling thirsty because plasma osmolarity has increased. Okay, and then you take water. Okay, and drinking of water, it is removing that thirst, or we can say it is causing satiation by removing the excitatory signal for thirst. Okay, but behind this, what exactly is happening? Okay, what exactly is happening, we have to discuss. Okay, however, the control system for osmoregulatory thirst cannot be that simple in organization. Okay, just now I told you that because of increase in the osmolarity of the plasma, we used to feel thirsty. Okay, but why immediately after drinking water, why that satiation is felt? That means why thirst is suppressed. Okay, so it is not a simple mechanism. Okay, so you see that. However, the control system for osmoregulatory thirst cannot be that simple in organization because a substantial delay occurs between the time when water is ingested and the time when it appears in the circulation. So this point is very important. Okay, my point is that we feel thirsty because there is increased plasma osmolarity. That means it is in the bloodstream. Okay, but when you drink water, okay, when water is reaching only the stomach, okay, let's say the stomach before absorption of water, okay, there is satiation for thirst, or thirst is inhibited. Thirst is inhibited. It means that there is a delay, okay, substantial delay. That means water has to be absorbed through the wall of the digestive tract to reach the plasma, or to reach the blood, to dilute the plasma concentration, or we can say plasma osmolarity. Okay, so we know that we are drinking water, but it has not reached the bloodstream. It has not reached the bloodstream, but thirst is already inhibited. Okay, why this is happening? Because the blood the blood is not yet diluted, because the water we drank, okay, it has not reached the bloodstream, but thirst is already over, or already inhibited. Okay, so that means this mechanism is not, satiation of that thirst is not a simple mechanism. That means it is not because of dilution of the blood, but there are some mechanism which is causing satiation for thirst. Okay, thirst satiation. Okay, because it is impossible to have that thirst satiation until and unless water is reabsorbed through the wall of the digestive tract and reaching the bloodstream. So theoretically, we can say that it will take time, okay, or practically it will take time. So specifically, in both dogs and humans, it takes 10 to 20 minutes for ingested water to produce significant decrease in plasma osmolarity. So this is very clear. Okay. So to decrease plasma osmolarity significantly, okay, the water we have ingested will take at least 10 to 20 minutes. Okay. That means if we consider only dilution of plasma osmolarity will cause thirst satiation. It means thirst will be satiated only after 10 to 20 minutes. Okay. Thirst satiation will take place only 10 to 20 minutes. But we all know that immediately after drinking water, within seconds, thirst satiation is there. Okay. So that means there are some other mechanisms, okay, neuronal circuits, or you can say neuronal mechanism, which is causing satiation of the thirst that we'll see. Okay. Whereas thirst and water intake are terminated much more rapidly, you already know. Thus, an early stimulus must occur that signals the brain in anticipation of subsequent rehydration. So, an early stimulus must be there. Okay. That signals the brain in anticipation of subsequent rehydration. Okay. Such an effect resembles feed-forward reflexes found in the control of numerous autonomic functions, like, example, insulin secretion in response to the taste of food before its digestion and assimilation. Okay. When you just taste the food, okay, in that condition, even insulin, okay, from the pancreas will be stimulated. So somewhat like that, in case of thirst satiation also, there are some neuronal circuits, okay, which will signal the brain to suppress the thirst center that we'll see. Okay. So under this, let's see first pre-absorptive inputs. Pre-absorptive means before absorption of the fluid you have ingested. Okay. So, you see, drinking water rapidly inhibits neuronal calcium activity in SFO thirst-related neurons even before plasma osmolarity or volume has been restored. Okay, so this is a neuronal signal. Okay, so SFO thirst-related neurons, okay, they are rapidly inhibited by drinking of water, okay, even before plasma osmolarity or volume has been restored. So within seconds, okay, these SFO thirst-related neurons, they are inhibited, okay. Input signals likely arise from receptors in pre-absorptive region which send projection to the brain. Okay, so there should be some receptors in pre-absorptive region. Pre-absorptive regions means the regions through which there is no reabsorption of water. Okay, that means we are talking about the digestive tract. Okay, receptors in reabsorptive regions. Okay, digestive tract through which no reabsorption takes place, but they are supplied with some nerve which can send signals to the brain. Okay, likely pre-absorptive inputs can be divided into number one, predictive cues, number two, oropharyngeal inputs, and number three, esophageal and gastric inputs. Okay, so we'll discuss one by one. Okay, so number one, predictive cues. Okay, so this is experimental. Okay, means experimentally it is said that you can see this one, placing a water bottle in the cage of 30 mice rapidly inhibits calcium activity in vasopressin expressing neurons in the supraoptic nucleus even before drinking has commenced. Okay, so suppose you just place a water bottle, okay, in front of a cage, okay, and that cage is having mice. Okay, so after just seeing the water bottle, okay, there will be inhibition of that we have already talked, calcium activity in the what to say, supraoptic nucleus even before drinking has commenced. Okay. So we all know very well that this psychological, we can say, psychological input is very important to control so many, what to say, physiological processes inside the body. Okay. So just by smelling, we know that just by smelling some food, if you are feeling hungry, so salivation will take place. Okay, just like that. Okay. Here also you see, just by placing water bottle in front of the mice. Okay. That will send signal to the brain, inhibits calcium activity in the supraoptic nucleus. Okay, this can be, what to say, inhibit the HERS. Okay, this suggests that these neurons receive rapid signaling about what are predicting cues, perhaps via visual or olfactory inputs, by seeing or by smelling. Okay, we can say, okay, they are sending signal to the brain to suppress, what to say, HERS. Okay, it prepares the body for drinking and anticipate the amount of fluid required to restore homeostasis. Okay, so this is predictive cues. So number B, okay, pre-reabsorptive inputs under this one. Number 2, oropharyngeal inputs. Okay, inputs from oral cavity and pharynx. Okay, inputs of water. Okay, let's say in the oral cavity and pharynx also signal fluid satiation. Let's see, gargling water can inhibit the sensation of thirst in dehydrated human for about 15 minutes. Okay, suppose you are feeling thirsty, but you just do the gargling of water. Gargling, we all know. Okay, these days also it is very common because of this COVID-19. Okay, so gargling of water, not drinking of water. Okay, it can inhibit the sensation of thirst. Okay, here around in case of human being for about 15 minutes. Okay, so this suggests that oropharyngeal receptors, nerve receptors, okay, they are present in the oral cavity and the pharynx. Okay, and these receptors, they mediate transient sensation of fluid satiation. Okay, that means they are sending signal to the thirst center, okay, to inhibit the sensation of the thirst. Okay, so gargling can also inhibit the thirst. Okay, this is not drinking. That means gargling means it is in the oral cavity as well as in the pharyngeal region. Okay. Oropharyngeal signal communicates to the brain only the volume of fluid ingested and is not its composition. So it is not saying that whether the solution, it is what to say, fresh mineral water, or it is having, what to say, glucose solution, or it is having chloride solution. Okay, so there is no difference. It is just detecting the volume of the fluid which is gargled, okay, not the composition of the solution. Okay, so oropharyngeal impulses signal for brain neurons. Okay. This mechanism allows the brain to track fluid intake in real time, thereby quench thirst, inhibit the secretion of ADH in anticipation of water absorption into the bloodstream. So actually, this mechanism can detect how much amount of fluid is to be, or how much volume of fluid is to be ingested, apart from satiating the thirst. Okay, so that means it can inhibit secretion of ADH in anticipation of water absorption into the bloodstream. That means that even before it is reabsorbed into the bloodstream, ADH secretion can be suppressed in anticipation of water absorption. How much volume of water will be absorbed, it can detect. Okay, so both water and hypertonic saline decrease thirst, suggesting that the type of fluid does not change the sensation of fluid satiation, at least initially. So as I told you, it is not detecting that type of fluid, whether it is pure water, mineral water, or hypertonic saline solution. Okay, both of them they satiate thirst, or they supply, detecting the concentration of the the fluid or the type of solution, but they are detecting the volume. Okay. However, hypertonic saline relief occurs only transiently with the sensation. There is a problem. Just excuse me. Good. Hello?
Yes, sir.
No, there was. It's visible or not? Slide.
Yes, sir. Visible. Sir.
So here again, however, oropharyngeal cavity does detect a difference between fluid and food. So this point is also important. Okay. Oropharyngeal cavity, the receptors which are present in the oral cavity and the pharynx, they detect a difference between fluid and food. Okay, that is why with fluids providing a rapid inhibitory signal to thirst, but not food. Okay, so that means that suppose you are chewing something, food. Okay, that will not satiate the thirst. But if you do some gargling and all with water, okay, that means there will be inhibition of the thirst. That means these oropharyngeal receptors, they detect difference between fluid and food. Okay. Cold temperature in the mouth, such as ice chips or a cold metal spout, are also experienced as more thirst quenching than warmer temperatures, suggesting temperature receptors in the oral cavity regulate fluid satiation. So I think we know very well this one. Okay. If you drink warm water when you are feeling very thirsty, if you drink some warm water or not cold water, okay, you have to drink lots of larger volume because the temperature is somewhat high. Okay, so that will not satiate the thirst center thirst easily. But if you take cold water, okay, cold water, okay, that will quickly, what to say, suppress the thirst. This we all know very well. Okay, so that means these temperature receptors in the oral cavity, they are also important in satiating the thirst. Okay, if you compare with that of the warm water. Okay, so the third one is that in post-absorptive, pre-absorptive inputs, the third one and the last one is esophageal and gastric inputs. Now, till now, we have discussed only up to the pharynx. When the fluid is reaching the pharynx, we know that thirst satiation is there. Okay, now you exactly drink water now. Okay, that means it will pass through the esophagus and then it will reach the stomach. Then what will happen here? The ingestion of water and hypertonic saline resulted in the rapid inhibition of subphonical organs, neurons, okay, in the thirst center. Okay, if you drinking water or if it is hypertonic saline also, if it is sodium chloride solution, okay, immediately what will happen is that there will be inhibition of the thirst center, you can say, okay. SFO neurons may receive signal from the gastrointestinal tract that depends on the osmolarity of the ingested fluid. Okay. So here the important thing is that what is the osmolarity of the ingested fluid? But as I told you, if it is pure water or if it is hypertonic saline, okay, immediately there will be inhibition of the third center. Okay, but you see that after some time, this osmolarity of the ingested fluid, it is important. Okay, changes in gastrointestinal osmolarity but not distension are sufficient to sustain thirst. Okay, so changes in gastrointestinal osmolarity. That means, suppose you drink, we are drinking water. Okay, osmolarity of the gastrointestinal tract, it will be decreased because you are drinking water. It will dilute the osmolarity of the gastrointestinal tract. Okay, so this is important and not the distension. Okay, when you drink water, there will be distension of the stomach. Okay, but it is said that that distension of the stomach is not sending any signal to the brain to inhibit the thirst center or to inhibit thirst. So it is said that gastrointestinal osmolarity or osmolarity of the stomach, okay, stomach fluid or gastric fluid, it is important to satiate the thirst. Okay, osmolarity is precisely measured within the gastrointestinal tract and then communicated to the thirst neurons in the brain. So depending upon what is the osmolarity of the stomach content, okay, thirst will be inhibited. Now those are the pre-absorptive inputs. That means before absorption of water into the bloodstream. Okay, now let's see the post-absorptive inputs. So we know that after drinking of water, it is reaching the intestine and large intestine also. And we all know that reabsorption of water takes place in these regions. Okay, in the longer term, fluid homeostasis is restored due to absorption of fluid into the systemic circulation. Okay, so we know that thirst is felt because of increased osmolarity of the plasma. Okay, so that is ultimately that plasma osmolarity, it should be decreased. It should go back to its normal value. That means the plasma should be diluted. So this can be accomplished because of reabsorption of water only, because of reabsorption of water and adding of water to the bloodstream only or to the systemic circulation. So in pre-absorptive input, we have discussed about some nerve signals, okay, which is causing satiation of the thirst. Okay, but those are, what to say, let us say that it is not a long-term satiation until and unless the water ingested is reaching the systemic circulation. So quickly or soonly, we can say, feeling of thirst will come back. Okay, so those pre-absorptive inputs, they are just for the temporary inhibition of the thirst. Okay, and after drinking of the water, water should be reabsorbed through the wall of the digestive tract and it should reach the systemic circulation. It will dilute the plasma concentration or plasma, and plasma osmolarity, it should go down, so that permanent, or we can say permanent satiation of thirst will take place. Okay, before dehydration takes place again. Okay, this is you see, in the longer term, fluid homeostasis is restored due to absorption of fluid into the systemic circulation and subsequent reduction of plasma osmolarity. This is detected by post-absorptive signals such as baroreceptors. Okay, baroreceptors you already know. Okay, pressure receptors and osmoreceptors, you know, they are there in the brain as well as in the kidney. And sodium sensors, they are mainly present in the subcortical organ. These signals prevent or diminish further fluid and salt intake. Okay, so these receptors, they are important, okay, in preventing or diminish further fluid and salt intake. So when sufficient amount of water has entered the bloodstream, okay, so these receptors, they are important in preventing or drinking of water. So for example, increased BP inhibits drinking, whereas lowered BP enhances drinking. Okay, so post-absorptive signals also includes renin-angiotensin aldosterone system, which promotes water and salt intake under condition of hypovolemia. So this is just a repetition. So another peptide or protein is atrial natriuretic peptide ANP released from the cardiac muscle under conditions of hypervolemia. ANP can inhibit fluid intake, suggesting a role in fluid satiation. So ANP is also important in satiating the inhibiting the fluid intake. That means the meaning of the post-absorptive input. Post-reabsorptive input means that when the fluid has already entered the bloodstream, okay, it stimulates some receptors that we have already discussed. The receptors, all those things, okay, osmoreceptors, so that further intake of fluid will be stopped. Okay, so this is for today. Thank you.
Thank you, sir. Thank you, sir.
Okay.
Thank you, sir.